My name's Ellen Carrick and I'm a PhD student at the University of Bristol in the Department of Physics. I study soft matter specifically, and in particular we're looking at the structural and colloidal stability of proteins. So in our lab, we are using the SuperDSF to investigate the structural stability of proteins in different formulation conditions. Part of my project is looking at the self-assembly of proteins. This has really wide biological implications because if we think about how our cells organize, we have our cytoplasm and we have organelles, some of which are membrane bound, like our mitochondria, but some of which aren't membrane bound. So then you're asking the question, how do two liquids kind of organize themselves? And the answer is biomolecular condensation. Biomolecular condensation is mediated by proteins that are intrinsically disordered. So they don't fold and form a stable kind of secondary or tertiary structure. They're actually more like long, elongated polymers. So we're looking to understand how intrinsically disordered proteins interact with each other. So it has implications in cellular biology, but also in the pathway of certain diseases such as Alzheimer's, which are associated with your tau protein in your brain that can misfold and aggregate. A lot of pharmaceutical companies are looking to branch out into biologics. So these are drugs that are essentially designed by biology. So they're protein-based drugs. And they have a lot of kind of unique and quite difficult challenges when it comes to making them stable in solution. So we use the SuperDSF to assess the structural stability of those proteins. So in our lab, we're looking to better understand kind of the building blocks of life, which are proteins. So we use techniques such as spectroscopy and some kind of protein purification techniques to better understand how proteins interact with each other and their structural stability. And that has kind of implications for everyone, really. If you have Alzheimer's or cataracts, that's your protein misfolding and aggregating. Really, we're trying to better understand the human body, but through physics. Proteins are really complicated molecules, so we need to find kind of the ionic strength and the pH conditions in which they are most stable. That's what makes the SuperDSF kind of the perfect instrument for us, because we're able to run a 384 well plate in a matter of seconds, really, which is so much faster than any other kind of plate reader we have in the lab. The SuperDSF generates tons of data from both thermal melt and chemical melt. Through the software, you can fit your data and generate delta G and delta H values within the software. So they're essentially ready for publication. So we were using the SuperDSF to assess the stability of IgGs. These are proteins that are produced naturally by the immune system to fight infection, but they're also a target for pharmaceutical companies to see if they can engineer specific IgGs to target specific diseases. If we're going to use them as medicine, we obviously need to make them structurally stable. To investigate the structural stability, we were running plates with those IgGs in different formulation conditions, so adding in different excipients that promote the structural stability of those IgGs. So with the SuperDSF, we were able to identify the best excipients to maximize the structural stability of those IgGs. The SuperDSF is a bright pink box in your lab that can run a 384 well plate in about 10 seconds and generate more data than your average plate reader could in about 24 hours. So it's our go-to tool for assessing kind of the structural stability of proteins. I would describe the SuperDSF as fast. It really is really quick. It's kind of amazing to see how quickly it can run a plate. Everyone that I've spoken to at Protein Stable has been really helpful and really quick at responding if there's anything that's come up that kind of needs to be addressed.